Hello everyone, once again I welcome you to my series of lecture, that is Understanding Pharmaceutical Science with Dr. Hariharan. So today's lecture we are going to discuss about the production of monoclonal antibodies. So the production of monoclonal antibodies can be done with a technology called Hybridoma Technology. So this lecture I have divided into three major segments. So first we understand about the monoclonal antibodies and hybridoma techniques as an introduction part. Second, we understand the principle of hybridoma technology. And the third part we will discuss about the production of monoclonal antibodies, that is production by hybridoma technology. So first we understand about the antibodies and the monoclonal antibodies and hybridoma technology. So we know already. So antibodies are the otherwise called as immunoglobulins or proteins produced against the specific antigen. So what happens naturally is once a person encounters a foreign substance, generally we call it as an antigen. So once it encounters it, it overcomes the innate immunity, then it enters into the blood. So once in extracellularly, what happens is this antigen against humoral mediated immunity gets stimulated. So one of the major cell plays an important role in the humoral mediated immunity is the B lymphocyte. So this B lymphocyte gets stimulated and activated, converted to an antibody secreting cell called plasma cell. So it secretes antibodies. This is called as immunoglobulins. So this antibody interacts with the antigen and helps in the elimination of this antigen. So this is about an antibody. So now it comes here another name called monoclonal antibody. In short form we call it as MAb. So MAb are the particular type of antibodies which is effective against a specific epitope of an antigen. So the antigen has a antibody determining site, a part of its sequence of protein which is called as epitope. Against that the antibody will get produced. So if one particular epitope, if antibodies are produced, then it is called as a monoclonal antibody. So these monoclonal antibodies are generally required by us in producing diagnostic kits like ELISA, in passive immunization, in drug targeting various ways. So we need to produce these monoclonal antibodies in large quantities. So monoclonal antibodies can be produced from the hybridoma cells through a technique called as hybridoma technology. So we have to produce these hybridoma cells. So how we can get these hybridoma cells and what is the science behind the development of this hybridoma cell, that we will discuss elaborately in this lecture. So this technology, this hybridoma technology was discovered in 1975 by two scientists, that is George Kohler from the West Germany and Cecil Milstein of Argentina. So both the scientists individually has developed this technology. So first we understand about the Kohler Experiment. So if you see his experiment in Kohler in 1974, he successfully produced a hybridoma cells. So hybridoma cells he produced by means of fusing a P3 myeloma cells. This cells is resistant to azaguanine with a lymphocyte, that is the B lymphocyte, which is obtained from the spleen of the mouse. So he has fused it, he has successfully fused these myeloma cells and these lymphoma cells to got a hybridoma cell. So hybrid means the joining of two characteristics. So he got this lymphoma, that is the lymphocytes, by immunizing against the sheep red blood cells. So he has injected the red blood cells of the sheep into the mice. So the red blood cells carries the surface protein which behaved as an antigen to produce these lymphocytes. So he fused these lymphocytes very successfully. By producing this hybridoma, he got two characteristics, but that we can tell that this hybridoma retained both the characteristics of the cell, that is the immortality, cell division ability of a cancer cell, that is the myeloma, and the antibody producing characteristics of a lymphocytes. So this hybridoma retain both the characteristics. So if this can be explained with a simple definition. If you see what is an hybridoma technique. So hybridoma technique is a technique or a method of producing a pure and uniform antibodies, that is immunoglobulins, against a specific target antigen. So this can be produced by the formation of the hybridoma cell, that is a hybrid cell of a cancer cell myeloma and an antibody producing cell lymphocyte, which is obtained from a particular donor. So this is called as hybridoma technology. So hybridoma technology is the joining of the myeloma with a lymphocyte. So then it produces the antibodies. Then this antibodies, what we need is a monoclonal antibody. So if you see the definition, what is a monoclonal antibody? So monoclonal antibodies are the products obtained from the hybrid cells by the hybridoma technology. So this hybrid cells is formed because of the fusion of myeloma cells and the lymphoma cells, which has an ability to divide fastly and produce a particular type of antibody, that means a particular unique antibody with one clonal, single clonal against a specific antigen is called as the monoclonal antibody. So the second part of our discussion is the principle of this hybridoma technology. So if you see the principle of the hybridoma technology, the basic idea of this hybridoma technology is to produce a lymphocyte and immortal. So production of immortalization of this lymphocyte is the core of this technology, which can produce an antibody at a continuous process, continuously. So how we got this immortalization of this lymphocyte is by the fusion of this lymphocyte with a non-antibody producing an immortal cell, that is a myeloma. So this I have explained with a diagram, if you can see. So what we are doing is simply we are giving an antigen against which a monoclonal antibody should be produced to an animal. For example, we have administered to a mice. So the mice will produce an antibody. So against this antibody, this mice will produce an antibody through the stimulation of the plasma cell, that is the B lymphocytes. So what we are doing is we isolate this B lymphocyte from this mice. Then we fuse with a cancer cell, that is a cancer myeloma cell. So we got a hybridoma cell. So this hybridoma cell retains the characteristic of immortalization, and it also retains the characteristic of production of antibodies against the specific antigen which has been immunized. So we get a large quantities of this monoclonal antibodies. So now we comes to the final part of our discussion, that is the production of monoclonal antibody, and that is the main part of our lecture today is. So if you see the monoclonal antibody production process, it has many steps and each steps are very important. If any problem occurs in a single step, then the final product won't be that much monoclonal in nature. So if you see the steps of monoclonal antibodies, the first step is the getting a purity and pure form of immunogen. Second is a selection of a perfect myeloma cell line which can form a good hybridoma cell. Third is a choice of an animals which can yield us a lymphocytes in large quantity. So fourth is the immunization of that particular pure form of immunogen to that animal. Fifth is the sacrifice of this animal and the isolation or separation of the lymphocytes. And the sixth step is the fusion of these lymphocytes with the myeloma cell, that is producing a immortalization of this lymphocyte cell. So finally, out of these fused cells, selection of the hybridoma cells which can be produce a good antibodies. So final next step is the isolation of the monoclonal antibody from this hybridoma cell and large scale production of this monoclonal antibody. These are the steps which is needed to get a monoclonal antibody production in large scale. So this I have illustrated with a diagram. If you see, First, what we are doing is we are administering the antigen to the animal. In this case, we have taken a mice. So, we are administered an antigen, otherwise we can call as a pure form of immunogen, against which the monoclonal antibodies should be produced. So, we administer it to this mice. Then what happens is it will produce the lymphocyte cells, that is the lymphocyte cells will be produced into the spleen. So, next we will isolate the lymphocytes from the spleen and we will keep it separately. Then we will select a myeloma cells. The myeloma cells generally should not carrying the HGPRT gene. So, HGPRT minus myeloma cells is taken. Then the next step, both the things has been fused with the help of a chemical called polyethylene glycol. This polyethylene glycol helps in the fusion of this myeloma cell with the lymphocyte cell. So, next step what we are doing is we have to need this hybrid cell, that is the hybridoma cells, which doesn't need the myeloma cells in pure form and the lymphocyte. So, we have to identify we have to select the hybridoma. So, what we are doing is we culture in a hat medium. So, hat medium is Hypoxanthine-Aminopterin-Thymidine medium. So, in this what happens is only the hybridoma cells will grow, the other cells will die. Then we will check the supernatant for the antibodies. Then each clone is taken separately, then the positive cultures are grown separately. Similarly, we check the antibody titer level of each clone and the characteristics of the antibody, whether it is monoclonal or polyclonal is checked. Then the high yielding strain positive clone is expanded. It is grown in large quantities either by in vitro through a petri disk in a lab or in vivo in an animal, so that will produce the monoclonal antibodies in large quantities. Then it is harvested and it is used for our purpose. So, if you see in depth of the each steps how it is done, if you see the pure form of immunogen, that is the purity of the immunogen, because the immunogen which is in pure form will have a single epitope. So, against the single epitope a single monoclonal antibody can be produced. Otherwise, if the immunogen is impure, then the polyclonal antibody can be the final product. So, the purity of the antigen is very important. So, you already know that immunogen is nothing but a chemical which produces a immunity. So, the pure form of immunogen will carry a particular antigenic determinant site called as epitope, through which the monoclonal antibody can be produced. So, if the immunogen characteristics plays a crucial role, if the molecular weight of the immunogen is more than 1000 Dalton, that is 1 kilo Dalton, it can produce a good immunologic property. If it is less than 1000 Dalton, then it should be coupled, it should be attached with an immunogenic molecule. So, generally an aggregated or a particulate antigen can produce a stronger response, whereas this low molecular weight will produce a lower response, becomes a poor immunogen. So, it can be coupled and can be converted to produce a stronger response with a carriers such as either an albumin or foul gamma globulin or keyhole limits hemocyanin or any synthetic peptides. So, this should be produced and kept aside for the administration for the next future steps. So, second one is the selection of the myeloma cell line. So, the cancer cell line should be selected in such a manner that it should only be ability to bind with a lymphocyte only, but this myeloma should not fuse in between this hybridoma cell. That means a myeloma lymphocyte can bind with an hybridoma one. So, this should not be allowed, it should only bind with a normal lymphocyte. And second one is the myeloma cell line used itself not have a property to sensitize the antibody production, because the cancer cells has an ability to produce the antibodies against it. So, this myeloma cell lines what we are selecting is that it should not have an ability to itself produce an antibody. If it produces an antibody itself against it, the antibody is produced by the lymphocytes, then we get a mixture of antibodies, then our product will be finally not useful. Then the third one, which is the most important one, is this: the myeloma cell line should be devoid of the gene HGPRT. So HPRT or HGPRT gene should be absent in this myeloma cell line. So because that is important for the selection of the hybridoma culture, right? So then the third step, that is the choice of the animal. So the choice of the animal depends upon the animal strain, which can gives a large quantity of lymphocytes through a spleen. So then moreover, it should not produce antibodies against the myeloma cells. So it have to ability to stimulate, immunologically get stimulated against that particular immunogen. So generally the mice is most preferred because it can be easily handled, the immunization can be easily done, and murine myeloma cell lines also available. So that's why the mice is preferred, so that we get the mice lymphocytes can be easily fused with the murine myeloma cell lines. So the strain which is generally selected for mice is a BALB/C strain, so which have a myeloma cell lines which can be easily fused with this, right? So next, the fourth step is the immunization procedure. So generally the antigen, that is the immunogen, can be injected either subcutaneously, or the most preferred route is the interperitoneally. That means administer peritoneal cavity of the animal, either alone, otherwise with the adjuvants. So it can gives a strong immune response. So mostly for mouse, a dose of 1 to 50 milligram per injection is repeated 2 to 3 times, which is sufficient enough to produce a strong immune response. So if the antigen is soluble in nature, then the first injection should be given in presence of an adjuvant, whereas the further injection can be in aqueous solutions. So generally for this type, generally the dose administered is 20 to 100 gram intraperitoneally, then given intravenously in the subsequent days. So it is incubated for 10 to 14 days, that is about roughly around 2 to 3 weeks. So the animal has an ability to produce enough titer value of antibodies against that particular antigen. So it is determined whether it is producing antibody or not, whether the antibody produced is against that particular antigen or not. So it is determined. Then once it reaches a higher antibody titer value after the administration of last booster dose, then the next step is carried out, that is the sacrification of the animal and the separation of the lymphocytes. So after the final dose, the animal is sacrificed, then the spleen is isolated, then the lymphocytes are slowly drained out along with the red blood cells. Then the lymphocytes are isolated or purified or separated through a gradient centrifugation technique, and it is taken away. So now we have cell lines, and also we have a B lymphocyte. So the next step is the fusion of this lymphocytes and the myeloma cells, that is immortalization by fusion with the tumor cell lines. So the fusion process between the myeloma cells and the spleen lymphocyte is done with the chemical called polyethylene glycol, especially PEG 1500. So in previously, when the polyethylene glycol identified, the fusion can also be done with the SEDI virus. So both are fusogenic agent, and finally it leads to the fusion of the cell. So if you see, the condition for the fusion process is that we need the following conditions should be strictly followed for getting a perfect fusion of this myeloma and the lymphocytes. So the first one is the myeloma cells which is selected for the fusion should be in the log phase, that is the exponential phase of its growth cycle should be selected, the first one. And the second one, the ratio of fusion between the two cells is that 1 myeloma cell against 2 to 5 lymphocytes should be there. And the third one is the 40% PEG can be used. So any concentration is slightly greater than can cause us the cell toxicity. So finally the fusion is carried out at 37 degrees Celsius at 7.5 to 8 pH. Generally 3 to 30 minutes it is carried out. Within 3 minutes sometimes the fusion can happen. So the fusion process occurs by two distinct stages. The first one is the cell agglutination and second is the osmotic cell swelling and heterokaryon formation. So if you see the cell agglutination, what happened in this stage is the myeloma cell and the lymphocyte cell membrane, plasma membranes will come close proximity and a cytoplasmic bridge between these cells will be formed. Then it favors the next phase, that is the osmotic cell swelling and the heterokaryon formation. That is, in this the polyethylene glycol will help in the induction of this cell fusion process. So this mixture of cells is exposed to these agents and it is maintained at an appropriate concentration so that the cytotoxicity doesn't occur. So within a minutes of exposure, it generally makes this plasma membrane to started getting merged together. So it merged together and forms a intercytoplasmic communication begins within 1 to 3 minutes and the dilution of the PEG will started occurring. So cytoplasmic mixing is done around 37 degrees Celsius for nearly up to 4 hours, but the cell clumping will be maximum in 30 minutes and the cytoplasmic mixing will almost complete within 40 minutes. So once this joins, so we get generally 3 types of cells in this one. One will be a hybridoma cell, that is the fused cell. One will be an individual lymphocyte. One will be a myeloma cells. But what we need is only the hybridoma cell which carries both the characteristics. So the next step is the selection of the hybridoma cell. So the entire technology of this hybridoma technology depends upon the type of the myeloma cell line and the selection of the hybridoma cell. So the selection of the myeloma cell should be devoid of this HGPRT gene, that is hypoxanthine guanine phosphoribosyl transferase gene. So this one not able to grow into a selection media, that is the HAT medium. This gene is absent means that cells cannot grow. So whereas the lymphocytes can grow and the fused cells can grow. So that is the first basic science between this. Why? Because of this the HGPRT gene is responsible for the DNA synthesis. So if you see any cell want to survive means it has to produce DNA. Nucleotides. So the DNA synthesis of nucleotides should happen. So the DNA synthesis of nucleotide can happen by 2 pathways. One is called the purine salvage pathway and second is called the de novo pathway. So in purine salvage pathway we need an enzyme called HGPRT. So it is needed for the production of DNA synthesis or production of DNA nucleotide by this pathway. If the HGPRT gene is absent, so in this case like the myeloma cell, it can synthesize the DNA by the another pathway called de novo pathway. So this de novo pathway can be blocked by this toxic media which helps in the selection of the hybridoma cells. So for selecting the hybridoma cells, what we will do is after the fusion we will put all the cells into a petri dish containing the medium called the HAT medium, that is Hypoxanthine-Aminopterin-Thymidine medium. This medium will help for the selection of only hybridoma cells, but due course it will be toxic to myeloma cells and the lymphocyte will be gradually die afterwards, because it divides very slowly. So this medium will specifically eliminate the myeloma cell and it allows the growth of hybridoma cell and the lymphocytes, whereas the lymphocyte will die eventually. So if you see the principle of this HAT medium, how this HAT medium works, helps in the selection of this hybridoma cell is that the HGPRT negative myeloma cells will generally die off in the HAT medium. So how it dies in the HAT medium if you see is that the HAT medium carries 3 basic chemicals. One is hypoxanthine, second is aminopterin, third is thymidine. So hypoxanthine and thymidine are essential for the synthesis of DNA through purine salvage pathway, whereas the aminopterin blocks the de novo pathway through the inhibitory activity against the dihydrofolate reductase. So, if a enzyme HGPRT is present, then it can utilize the hypoxanthine, then it can forms the DNA by the I already told that for analog pathway, that is the purine salvage pathway, whereas in this case, if the HGPRT is negative, then it has to synthesize the DNA through the de novo pathway. So in the HAT medium, we are adding the aminopterin, which is an analog of the folic acid. So because of this, it blocks the enzyme dihydrofolate reductase. So by blocking of this enzyme, what happens? The de novo pathway doesn't occur. So since the de novo pathway does not occur, the myeloma cells cannot survive. It will eventually die. So whereas the hybrid cells between the myeloma and the lymphocytes. So the lymphocyte carries the positive gene, that is the HGPRT positive gene. That means it has an ability to synthesize DNA through purine salvage pathway. So it produces the DNA, it will survive. So both the hybrid cells and the lymphocyte can survive in the HAT medium. But what happens is that the lymphocyte will slowly grows, whereas the hybridoma cell, because of cancerous nature, it grows faster. So it eliminated by the poor growth during the in vitro. So if you see graphically how the inhibition of this de novo pathway occurs is that the aminopterin, which is an folic acid analog, blocks the enzyme dihydrofolate reductase, which is necessary for the synthesis of stimulation of the thymidylate synthase through methylene tetrahydrofolate. So if the thymidinate synthase is present, this only converts the deoxyuridine monophosphate to deoxythymidine triphosphate. So this pathway gets blocked. So the de novo pathway of DNA synthesis is blocked. So the myeloma cells will die and the lymphocyte will die because of its poor growth. Only the medium will favors the growth of the hybridoma cell. So we get the hybridoma cell. So the next step is the isolation of the monoclonal antibody producing this hybridoma cell. What happens is that in the HAT medium, all the fused cell will grow, but all the fused cell doesn't produce the monoclonal antibodies. Some will produce a polyclonal mixture because of its sensitization during the process. So the single antibody producing monoclonal antibodies should be isolated and grow individually. So what we will do is we will slowly dilute this suspension and the positive clones are slowly isolated, then it is grown separately, then its antibody titer level is analyzed until the type of monoclonal antibody is produced is checked. Then this transfer to a fresh media for growth until a consistent monoclonal antibody producing hybridoma clone is selected and it is examined and finally stored. Then the final step is the commercial production of these monoclonal antibodies. So we generally store this identified monoclonal antibody strain into a cryopreservation. Generally we call as a liquid nitrogen storage system. Then it is cultured whenever we required it. So we produce a large scale like I have illustrated in the figure. So this large scale production of monoclonal antibody is done through a bioreactor. So in this we will keep these hybridoma cells into a medium. So then it is grown provided an optimum temperature, pH and concentration of oxygen, everything. So it releases the antibodies. So this antibodies comes with other proteins also. Then by downstream processing, a pure antibody is isolated. So this is how a production of monoclonal antibodies is done. So I can simplify the steps once again to you. So the first one is what we are doing is we are getting a pure form of immunogen. Then we are administering this immunogen to the animal. So the animal, what happens is it produces the lymphocytes in the spleen. From the spleen it is isolated and we take the HGPRT minus myeloma cells, then it is fused with the in presence of polyethylene glycol to produce the hybridoma cells. Then the hybridoma is selected through a hat medium. Then the pure form of positive clones are subculture and the titer values are continuously calculated until a positive large large quantity producing high antibody titer monoclonal antibody producing hybridoma is selected. Then it is cultivated either by in vitro or in vivo to produce a large scale production of monoclonal antibodies. Then it is harvested and used. So thank you very much for patiently listening this production of Hybridoma technology. So in future lectures you can understand the various concepts of pharmaceutical biotechnology and the pharmaceutical science. So thank you very much.